Okay, before continuing, I thought we'd look at this picture and get an idea or get a refresher about where everything is at. So here we have a synaptic connection, and we have the axon bulb, which is this area that's highlighted in purple from the nerve terminal. We have the presynaptic membrane, which is in green. We have the postsynaptic membrane, which is in red. And we have the synaptic cleft, which is the space between the presynaptic and postsynaptic membrane. So the previous video went over all the stuff that I'm about to go over, but we're going to read through this and cover it step by step. Postsynaptic potential, PSP, and the axon initial site or initial segment, AIS. In the CNS, a neuron can receive inputs from thousands of other neurons through synaptic connections. And we have a picture here of a neuron. In this picture right here, all the little red dots are synaptic connections being made to this neuron's soma and dendrites. As you can see, there are a lot of them. And in the cartoon picture, it is also depicting the same thing, and it's showing you the axon initial segment, which is right here, right? And you can see the start of the axon with the myelin sheets right here. These synaptic connections are made at the soma and dendrites. The postsynaptic membrane contains, among other proteins, transmitter-gated ion channels and voltage-gated sodium channels. Note that the voltage-gated sodium channels on the soma and dendrites are not the same as the channels we encounter in the axon membrane. And we'll cover what these voltage-gated sodium channels do shortly. Transmitter-gated ion channels are either excitatory or inhibitory. Excitatory transmitter-gated ion channels are sodium or cation channels, which can be specific for cations with a single positive charge or all cations. So these cation channels that we're speaking of here, they can be specific for either all cations that have a single positive charge, or specific for all cations, meaning that even cations that have a +2 charge will go through along with the ones that have a +1 charge. When an excitatory transmitter-gated ion channel is activated by the binding of its neurotransmitter to its ligand binding pockets, the postsynaptic membrane is depolarized. The level of depolarization is dependent on the concentration of neurotransmitter in the synaptic cleft within a period of time, which itself is dependent on the frequency of action potentials being received by the presynaptic membrane. Note, this is why a higher action potential frequency is a stronger signal. So when you have a higher action potential frequency, you will release more neurotransmitter into the synaptic cleft, thus opening those transmitter-gated ion channels, those excitatory transmitter-gated ion channels, which will lead to that patch of membrane becoming very permeable to sodium or cations. Then, sodium will move down its electrochemical gradient into the cell, past the postsynaptic membrane, into the neuron that is receiving the signal, and that patch will become depolarized. If the membrane is depolarized a sufficient amount, voltage-gated sodium channels will open. This is a positive feedback loop that amplifies the excitatory signal. The greater the depolarization due to transmitter-gated ion channels activation, the greater the positive feedback loop by the voltage-gated sodium channels. Maximum depolarization is near sodium's equilibrium potential. So what do we have here? We're going to just draw it out, or write it out. We have neurotransmitters, and these are going to be in the synaptic cleft, which then go on to activate transmitter-gated ion channels. And these are excitatory in this case, so we'll just put a EX here for excitatory. And these are on the postsynaptic membrane. This leads to depolarization of the postsynaptic membrane, which leads to the activation of voltage-gated sodium channels, which has a positive feedback loop on more depolarization. Now, the thing to notice is that if any of this stops, everything downstream stops. So if neurotransmitters are not being released into the synaptic cleft, then this goes away. And if that goes away, this goes away, this goes away, and this goes away. So everything is dependent on neurotransmitter release. And where does a neurotransmitter come from? So the neurotransmitter is being released from the presynaptic membrane. And that release comes from depolarization of the presynaptic membrane. So an action potential will lead to depolarization. This is of the presynaptic membrane, which leads to the activation of voltage-gated calcium channels, which is also on the presynaptic membrane, which leads to neurotransmitters being released into the synaptic cleft. So again, if I go ahead and increase the frequency of action potentials, I increase the amount of depolarization of the presynaptic membrane. They will occur more frequently. It will lead to more frequent activation of voltage-gated calcium channels, which means they're opening more frequently. That means more neurotransmitters being released into the synaptic cleft, which means more activation of transmedicated ion channels on the postsynaptic membrane, which leads to more depolarization of the postsynaptic membrane, which means more activation of voltage-gated sodium channels, which increases the positive feedback loop on depolarization. Now, if we scroll back up, if the action potentials are of low frequency or if they stop, period. So let's go with that they stop. There are no action potentials being generated by the cell that is synapsing with this other cell. Then no action potentials, no depolarization of the presynaptic membrane, no voltage-gated calcium channels are activated, no neurotransmitters release, no transmedicated ion channels are activated, there's no depolarization, the voltage-gated sodium channels are not going to be activated, and there will be not any positive feedback loop for those voltage-gated sodium channels. I would recommend that you learn to draw things out like this, and then see the path that is taken, and where anything that happens to this pathway, how it affects things downstream. So downstream will always be, in this case, depolarization, because this is the big event, the depolarization of the postsynaptic membrane. Okay, continuing. Therefore, an excitatory neurotransmitter is excitatory because the transmedicated ion channel it opens will lead to the depolarization of the postsynaptic membrane, or an excitatory postsynaptic potential, an EPSP. Inhibitory transmedicated ion channels are potassium or Cl- channels. When an inhibitory transmedicated channel is activated by the binding of its neurotransmitter to its ligand binding pockets, the postsynaptic membrane is hyperpolarized. This is due to the efflux of potassium, or the influx of Cl-. The level of hyperpolarization is capped at potassium or Cl-'s equilibrium potentials. The level of hyperpolarization is dependent on the concentration of neurotransmitter in the synaptic cleft in the period of time, which itself is dependent on the frequency of action potentials being received at the presynaptic membrane. Note, this is why a higher action potential frequency is a stronger signal. The activation of inhibitory transmedicated ion channels has the effect of making the postsynaptic membrane, a patch of membrane on the soma or dendrites, resistant to depolarization. This change in membrane permeability and resistance to depolarization is called an inhibitory postsynaptic potential. So we can draw this out exactly the same way we had it before. We have neurotransmitter release in the synaptic cleft, which leads to the activation of transmedicated ion channels, and this is on the postsynaptic membrane. And this leads to hyperpolarization of the postsynaptic membrane. And this makes this patch of membrane resistant to depolarization. If we again do the same thing we did before, we can say that an action potential is received, and this is at the presynaptic membrane, and this leads to the voltage-gated calcium channels being activated, and again, these are at the presynaptic membrane too, and this leads to release of neurotransmitter into the synaptic cleft. So again, let me scroll this up a little bit. If I have an increase in action potential frequency, the voltage-gated calcium channels on the presynaptic membrane will open up more frequently, you'll have more neurotransmitter release into the synaptic cleft, and this leads to more transmitigated ion channels on the postsynaptic membrane to be activated, which leads to increased hyperpolarization of the postsynaptic membrane. Now, if the action potential stops coming in to the presynaptic membrane, this will stop, voltage-gated calcium channels cannot open, and neurotransmitter won't be released into the synaptic cleft, transmitigated ion channels will not open, and you will not get hyperpolarization of that membrane, that patch of membrane. And just going back, this depolarization here would be an EPSP, and this hyperpolarization here would be an IPSP. Therefore, an inhibitory neurotransmitter is inhibitory because the transmitigated ion channel it opens will lead to the hyperpolarization of the postsynaptic membrane, inhibition of that patch of membrane, and its immediate surrounding from being depolarized. The postsynaptic potential. Each individual synaptic connection to the soma and dendrites is either inhibitory or excitatory. So when we look at this picture right here, each of these individual synaptic connections could be excitatory or inhibitory, and they can't be both. So either they're going to hyperpolarize a patch of membrane or depolarize it, causing an IPSP or EPSP. The additive effect of these individual EPSPs and IPSPs will lead to an overall change in the soma's membrane's potential. So the soma right here that we're looking at, and the dendrites are kind of being shown, the stems of the dendrites are kind of being shown. Eventually, all these EPSPs and IPSPs will have an overall effect on the membrane potential of the soma. If the excitatory signals outweigh the inhibitory signals, the soma's membrane will be depolarized. The depolarization will spread to the axon initial segment, which is the area right here. The AIS is responsible for computing the frequency of action potentials being sent down the axon. If the PSP, the combined PSP, is past a minimum threshold, an action potential will be sent down the axon. And this goes back to the picture we had earlier about an action potential. Actually, let's go back and look at that picture. So what we want is for the potential at the AIS to get past this threshold right here. If it is past that threshold, then an action potential will be generated. Otherwise, if it's below that threshold, you'll have failed initiation, and you will not get an action potential. If the PSP is past threshold voltage, but relatively low, the frequency of action potentials will be low. If the PSP is past threshold and relatively large, closer to sodium's equilibrium potential, then the frequency of action potentials will be higher or near maximum frequency the neuron can fire action potentials. Okay, so we're going to take this apart really quick. And we can do it right here. So we have said that a higher frequency action potential is a stronger signal. So a neuron has a maximum frequency it can fire an action potential. So we have max frequency of action potential firing, but there's also a minimum frequency, meaning that this is the slowest it can fire action potentials. Okay? And you have all this space in between. So the signals coming in to the soma and the dendrites could lead to an action potential being at minimum or maximum or anywhere in between. Now, if the PSP is low but past threshold, so low PSP, you'll be somewhere in here. But if the combined PSP is high, then you want to be somewhere here. Because a high PSP means that there's a lot of excitatory signals coming into the soma and dendrites, and the neuron should compute a strong signal to send down its axon. And that strong signal will be in the form of at high frequency action potentials. Whereas if the excitatory signals are not that strong and you have some inhibitory signals coming in, then you'll have a low PSP. And if that PSP is past the threshold level, then you want to fire a signal down the axon that matches the signals coming in to the soma and the dendrites, so you would want a lower frequency of action potentials being fired. Basically, if you do not have this, for example, all sounds will be at the same loudness. You will not have low sounds being low and high sounds being high, okay? Or if you touch something, if you're being gently touched, you get a lower feeling of pressure. Whereas if you're being pinched or being hit hard, then you would feel it more. If this did not exist, then every signal past threshold will give the same response. And that is what's being shown here. For example, you have the combined PSP being shown here. You have it relatively lower in this picture, and you get an action potential frequency that looks like this. Whereas if the combined PSP is higher, then you get a higher frequency action potential. And what this graph right here in C is showing is that the frequency of action potential is firing. Okay? So here is the highest frequency being fired, right? And here is the lowest. So high, low, and the x-axis, the magnitude of the combined PSP. And once you're past threshold, you can see at lower combined PSP, the frequency is low, but it starts to increase as the PSP increases until it reaches that maximum frequency for action potentials being able to be fired by that neuron. If it's a fast-firing neuron, the high frequency will be higher. If it's a slower firing neuron, the high frequency would be a little bit lower, okay? And we're going to stop here and continue by how this computation is made, meaning how does a neuron at the AIS compute the signal coming into it from the soma and the dendrites, that combined PSP, and make the correct decision on what frequency to fire its action potential. Okay guys, thank you. Bye-bye.